In capillary fluid dynamics, we're looking at the movement of fluid between the capillaries and the interstitial spaces, or the spaces within the tissues, cells. So this is a balance between what's called filtration, the movement of fluid out of the capillaries, and reabsorption, that is the movement of fluid into the capillaries, and how changes in this dynamic of filtration and reabsorption can end up leading to edema. So let's first look at everything that's involved and get kind of an idea of what normally occurs without any disorders or imbalances that may occur. So capillary fluid dynamics involves, first again, filtration. That is the movement of fluid out of the capillary at the arterial end. And you can see here fluid is being pushed out of the capillaries. And then at the venual end, we're going to have reabsorption. That is, fluid is pulled into the capillaries. Now this may be the exact same fluid that left, may just quickly get reabsorbed, but we need a balance between those two. And so this movement of the fluid is going to end up allowing for regulation of fluid volumes both in the blood and the interstitial fluid. But again, we're going to want to make sure this is a balanced approach between filtration and reabsorption. So what is it that affects capillary fluid dynamics? Well, it's going to be dependent on two pressures. First of all, blood pressure. Blood hydrostatic pressure we can call capillary hydrostatic pressure or capillary blood pressure. That pressure is think of the blood being forced through the blood vessels, pushing up against the blood vessel wall, pushing fluid out of the capillaries. Whereas colloid osmotic pressure, or it's called oncotic pressure or capillary osmotic pressure, this is created by the presence of solutes or large molecules like the plasma proteins, which are the biggest contributors to osmotic pressures, particularly albumin. And since we have a lot of albumin in our blood, the blood is hypertonic compared to the interstitial fluids, which means we're going to pull fluid in. Remember, fluid's going to go from high concentration to low. Well, if I have a lot of solutes in my blood, I have very little water or fluids and a lot of fluids outside in the interstitial spaces. So that fluid's going to go from high to low or be drawn into the capillaries. So think of that as a pulling fluid into the capillaries, whereas blood hydrostatic pressure is a pushing out. And so net filtration pressure is basically the differences between those two. Whoever wins in a particular area of the capillary, be it blood pressure or colloid osmotic pressure, whoever's bigger is the guy who wins. So let's take an example of that here. At the arterial end, blood pressure is relatively high. It's at 32 millimeters of mercury. So that means we're pushing that blood up against the wall of the capillary, forcing fluid out into the interstitial spaces. Whereas osmotic pressure is at about 22 millimeters of mercury. That's pulling fluids into the capillaries because of all those plasma proteins, drawing it in. So who wins is who's bigger. Since blood pressure is bigger than osmotic pressure, then the net pressure is 10 out, the difference between those two, and that is we have in the capillary end, we have filtration. At the venual end, then, we've got a different scenario because blood pressure has dropped since we're farther away from the heart. So now blood pressure is only 15 millimeters of mercury. We still have the same plasma proteins in the blood causing causing the osmotic pressure. So that osmotic pressure is still at 22 millimeters of mercury. So now who wins? Well, osmotic pressure does because it's bigger. So therefore there's a net movement of fluid in. So the net pressure is minus 77. That is, fluid moves back into the capillary, and that's referred to as reabsorption. So arterial end, you end up with filtration, blood pressure wins. At the venual end, we end up with reabsorption because osmotic pressure wins. But notice it's not the same balance. So we have 10 millimeters of mercury pushing it out and only 7 millimeters of mercury pulling it in. So that means we accumulate a little extra fluid into that interstitial spaces, and we don't pick up all that fluid at the venule end. But we don't end up with edema because of our lymphatic system. Here you can see the blood's flowing through the capillaries, and at the arterial end you've got filtration pushing fluids out. Over here represents the venule end, which means fluid is moving in because of all the plasma proteins. So if there is any imbalance between those two, as in we saw in the previous slide, then these lymphatic capillaries can pick up that excess fluid and travel through lymphatic vessels until it arrives into your subclavian vein, where it dumps all the fluid into that subclavian vein to restore the blood volume. Now, if the lymphatic vessels become blocked, you can end up with a condition called elephantiasis. Now, this isn't something that happens here, so don't think your legs are going to swell up because you get this parasitic worm that loves to live in lymphatic vessels and block them. This disease is only found in Africa, but what it does is that worm ends up blocking the lymphatic vessel. So now the fluid imbalance, that is when there's more filtration than reabsorption, the lymphatic vessels can't pick up the extra fluid because they're blocked. And so the fluid accumulates and you get really severe edema. Now realize it took me a while to find this picture because I was trying to find one that was appropriate for class. And in other words, gentlemen, there's a lot of lymphatic vessels in the groin area, and so that tends to be the area that's blocked. So you can imagine what in turn becomes quite swollen or swells with edema. And so you can Google that yourself and get other pictures of elephantiasis. Now we can also get this lymphatic blockage too, say if a woman has a mastectomy, they often remove lymph nodes with that, and with the lymph nodes then being removed, there's blockage in the lymphatic vessels, and so you can see swelling occurring in the woman's arm because those lymphatic vessels can't drain the excess fluid that may accumulate in that arm. So therefore they usually put drainages into that area to help drain the excess fluid until the vessels are healed. Let's now look at normal capillary bed and how filtration and reabsorption interact, kind of in a graphic diagram here. And then we'll kind of mess with this diagram to see how we can affect filtration and reabsorption by altering pressures or plasma protein levels. So first, let's get oriented. On the x-axis here, this is the length of the capillary. Here is the arterial end of the capillary. Here's the venule end of the capillary. And then the y-axis is a measure of pressure. So we can think of it in terms of capillary pressure. That would be the blood pressure, which decreases as you move away from the heart. So you can see the blood pressure decreases as you go from the arterial end to the venule end. But osmotic pressures stay the same. That is, since we have the same concentration of plasma proteins in the blood the entire length of the capillary, osmotic pressure is not going to vary. So in this diagram, what we want to see is that in areas of the capillary where blood pressure is greater than osmotic pressure, you end up with filtration. And in areas where osmotic pressure or oncotic pressure, same thing, is greater than blood pressure, you're going to have reabsorption. As long as these two triangles are the same size, that means we have the same amount of filtration as reabsorption. So the loss from the plasma equals the gain to the plasma of your blood, and there's no net change. That is, the fluid gain and the fluid loss is the same. And so this would be kind of the normal capillary bed. Okay? So keep in mind, as long as these two triangles are the same size, filtration and reabsorption equal each other. But now let's imagine if we have an increase in blood pressure. So instead of having it at 35, let's say it's higher than that. So at the arterial end, the blood pressure is much higher, but at the venular end, we just kept it the same. Osmotic pressure, oncotic pressure, hasn't changed. Now there's a bigger length of the capillary where we see filtration going on because blood pressure is greater than osmotic pressure, so the push out is greater than the pull in, and we get lots of filtration, whereas there's a smaller length of the capillary, where we see reabsorption. So we have a lot more filtration than we do have reabsorption, so we end up losing more fluid into the plasma and not reabsorbing it. And the lymphatic vessel can't keep up with this huge excess of fluid moving out of the blood, and so therefore you end up with edema. Another example we can see is with a decrease in blood pressure. So now notice I've dropped the blood pressure at the arterial end, I've just left it alone at the venular end, and again, osmotic pressure hasn't changed. So here we have a smaller length of the blood vessel where we've got high blood pressure compared to osmotic pressure. So only this part of the blood vessel do we have filtration, but we've got a huge area that has reabsorption. So we end up with a lot more reabsorption than filtration. So in this case, then, the net gain is in the plasma. That is, the blood actually gains fluid. Now, in the case of shock, where blood pressure is very low, this is like giving the body an auto transfusion. That is, it's like hooking up your own internal IV. Instead of dumping fluids through an IV into circulation to try to restore blood pressure, we have fluid from our interstitial spaces being dumped into our blood to up blood volume and therefore help restore blood pressure. So that kind of is a nice little trick to help us out in cases of shock. Now let's look at another example. Let's say we have an increase in venous pressure. This can happen with congestive heart failure. Typically in congestive heart failure, one side or the other side of the heart fails. It's either right-sided congestive heart failure or left-sided congestive heart failure. Left is the more common type, since more heart attacks occur on the left side. The left side of the heart becomes bad at pumping blood, but the right side of the heart is unaffected. It pumps blood just fine. Now, if the right side of the heart is pumping just fine, then it's going to dump its stroke volume, say 70 mils, into the lungs. And that means 70 mils of blood returns to the left side of the heart. But the left side of the heart can't pump out 70 mils. It isn't as effective a pump. Maybe it can only pump out 40 mils. That means of the 70 mils that wants to come back, it only pumps up 40, which means I've got 30 left. And then in the next beat, I still want to bring 70 back to the heart, but the left side can only pump out 40 more. So you can see, kind of get a backlog of blood in the lungs. If I get this backlog of blood in the lungs, that builds up pressure in the lungs. So there's the increase in venous pressure that I'm talking about. It's this tie up of blood in the pulmonary veins, increasing the pressure, the venous pressure in the lungs. Okay, so that's the scenario that we have here. I've got normal arterial pressure at the arterial end, but I've got a higher venous pressure. So notice I have a lot more filtration than I do reabsorption. Since I have a lot more filtration, I end up having more fluid leaving the blood than reabsorbed back into the blood, and therefore again I get edema. So one of the more common symptoms of congestive heart failure is pulmonary edema, accumulation of fluids in the lungs. Now let's mess with oncotic pressure. Let's say there's a protein deficiency or tissue damage that results in the loss of plasma proteins. So notice now our blood pressure is normal, starts at 35, drops down to 15, but we've dropped oncotic or osmotic pressure here. So again, notice we get this huge amount of filtration, very little reabsorption, Anytime filtration is bigger than reabsorption, I end up with edema. Now, this kind of imbalance we can see with, again, protein deficiencies or starvation. If a person has been starving, they'll first use up all their fat stores, and then they're going to turn to their muscles and pull the protein out of the muscle. And when they've run out of muscle, they're going to start going at the plasma proteins, the albumin that's in the blood. If I don't have albumin in my blood, then I don't have as high an osmotic pressure. I don't have all those plasma proteins drawing the blood into the capillaries, and therefore that's that drop in osmotic or oncotic pressure that we see on this graph here. And so the result is edema, and that's why you see the swollen bellies in children with severe protein malnourishment in areas where maybe there's been a famine. Also, you can get that when there's capillaries have been damaged, like in severe burns, and the proteins in the blood, the plasma proteins or the albumin, leaks out of the blood into the interstitial spaces. So now I don't have those plasma proteins in circulation anymore, and then that could also then result in severe edema in those, particularly in the areas where the burns are, but it could even be systemic.